Welcome back to my mini exam video number 5. In this week's video, I will be going over the effects of the 4 types of point mutations on DNA sequences. In this visual, I have given the original DNA sequence with the top strand being the coding strand and the bottom strand being the template strand. Now I'll give the products of transcription from the DNA sequence. Now you can see I have given the transcribed mRNA from the DNA sequence coding strand, which is identical to the coding strand except for the thymine being replaced with uracil. The transcribed mRNA is also known as the wild type because it has 0 mutations. To translate this mRNA sequence, we will start at AUG because this signals the start codon for the translation. Then we have UGG, which translates to TRP, and then AUU translating to ILE, and then ACU to THR, and finally we have UGA, which translates to the stop codon to signal the end of translation. Now I'll go through examples of mutations that could affect the end product of the translated amino acid sequence. I'll describe a missense point mutation first. A missense mutation is a mutation in a single base pair that changes an amino acid in the final product of translation. In this example of transversion mutation has taken place because the purine A has been changed to the pyrimidine T. This single point mutation in the DNA sequence caused the mRNA sequence to code for PHE instead of its original intent of ILE. Mutation I will describe is a nonsense mutation. A nonsense mutation occurs when a point mutation in the DNA sequence causes translation to stop earlier than originally expected. In this example of transversion mutation has also taken place because the pyrimidine G has been changed to purine A. This single point mutation in the DNA sequence caused the mRNA sequence to code the stop codon UGA very early, so not all of the mRNA sequence ended up being translated to proteins. Mutation is a read-through. A read-through mutation occurs when a point mutation in the DNA sequence causes translation to go on longer than expected because the original stop codon has been changed. In this example, a transition mutation occurs because the pyrimidine G has been changed to pyrimidine C. This single mutation in the DNA sequence caused the mRNA sequence to continue coding because it was changed to being Sir codon UCA instead of the UGA stop codon. This translated in this example ended 2 proteins later when the next UGA stop codon was translated from the mRNA strand. Last mutation is a silent mutation. A silent mutation occurs when a point mutation is made in the DNA sequence, but this change does not actually end up affecting the translated end product. In this example, a transversion mutation occurred again because the pyrimidine T was changed to purine A. This single point mutation did not affect the translated sequence because the affected region is still translated to the protein ILE.